And well, I have a slight problem of sharing the screen. So Laura will pass the different slides on time. So the next one, please. We are talking today on oligodendrocytes. And as you know, this is a cell type that was described by one of the most important direct disciples of Cajal, Pío del Río Ortega. The next one, please. And Pío del Río Ortega did a work by himself in 1919, where he described that the third element of the central nervous system that was described by Cajal with the gold chloride technique was indeed formed by microglia and oligodendrocytes. And in this labor, he received just a few collaborations. One of them was from Bilder Penfield, that was working with Pío del Río Ortega in Madrid, and also by an Australian-born girl, that is Dorothy Russell, that was working with Bilder Penfield and then with Pío del Río Ortega in Oxford during the Second World War. The next one, please. This is just only the example of maybe the most successful of the disciples of Cajal. You have here the direct ones. And the particularity of these guys is that all of them, their main discoveries remain fully active 100 years later, what in science is not so easy. And among these disciples, there were some others. The next one, please, like Howard Florey or Laura Foster. Can you go back for a moment? That were born also in Australia and were that spent some time working with Cajal. In 1911, Laura Foster, and in the 1920s, Howard Florey, coming from Oxford. The next one, please. Then, after this very brief introduction on the history of neuroscience and the role of Spanish and also Australian researchers, I will focus in what I've been working for more than 20 years now, that is oligodendrogliogenesis and CNS myelination. We are relatively obsessed with this cell type, that is the oligodendrocyte precursor cells. That are the cells that they are born to run in the way that they colonize the entire CNS, both white and gray matters, and they follow very different ways to colonize the different structures in the CNS, and they use for this molecular mechanisms that are quite well known for neuronal migration and axonal guidance during development also. We focused, the next one please, a lot in the early oligodendrogliogenesis, but as also Ryan said us before, there is a large amount of myelin that is produced in the CNS in the late prenatal development, and especially in species like ours, in relatively old more time. I mean, it extends up to the 20 years old in the formation of myelin in the prefrontal cortex. And there are some regions, like for example, the cerebral cortex or the cerebellum, where the most dramatic change that you can see after birth is the dramatic increase in the white matter tracts and fibers that appear there. The next one, please. This is something that was very interesting for us because we already knew that OPCs, oligodendrocyte precursor cells, are also present in the brain of adult human and mammals, both from healthy people and patients. And it was not very clear which was the reason of the presence of these cells there. And 10 years ago, a couple of papers, one from Kailin Young in the lab of Bill Richardson, another one from Giulio Tomasi in the laboratories of Jeff Litzman and Paola Arlotta, did two discoveries that up to me are very, not only fantastic, but also very transforming in this field. Kailin Young saw that the incorporation of newly generated oligodendrocytes were not only to form myelin around previously not myelinated axons, but also in previously fully myelinated tracts, like, for example, the optic nerve. And the work by Tomasi and Marlotta so that very subtle changes in the pattern of myelination of similar neurons would impact very significantly the way of these cells to transmit electrical messages and, of course, to reinforce circuits and be a true actor of brain plasticity and physiological plasticity with no requested new formation of connections. The next one, please. In this context, we know now that the changes in myelination can be relatively subtle and not only related to the thickness of the myelin, but also the pattern of the myelination along the axons, also the length of the nodes of Ranvier and the presence of different ionic channels within the node and the paranodes, etc. And there is a continuous change of this scenario from early development, so young, mature brains, or in the upper right, you can see that in the older animals, there is again a decline of the myelination or the presence of myelin in these structures. And it is fascinating how we can, we have developed different, many data about the positive influence for myelination or myelin formation and bad influences. That is not the case here, but would be very interesting also to check in different circumstances. Isolation, social isolation, has been very dramatic during the pandemics and is one of the main negative influences for the formation of myelin during development and also in the adult brain. So the next one, please. In these conditions, since years ago, we have some friends that are present in these stories. One of them is the fibroblast growth factor 2, FGF-2, and sonic hedgehog. That there are plenty of different works describing this, but when you scratch in the literature, you see that there are very subtle data that do not confirm or fully confirm the previous observations, or it is a kind of slight changes of perspective, etc. Overall, we can say now that as well as for these old friends for other molecules, we more or less know which are the OPC, the molecules that are involved and the pathways that are involved in the OPC cell fate, also more or less in the survival, in the proliferation, and even in the motility or the migration of the OPCs to colonize the CNS. But the largest differences rely on the role of these molecules and pathways in OPC differentiation and formation of myelin, especially in the different scenarios where it can be formed. The next one, please. Regarding this, some years ago, we generated a transgenic mice to study anosmin-1, that is a protein that is highly conserved in the phylogenetic scale, but that the gene encoding this, that is CAL1, or the former CAL1 that is known as ANOS1, has not been identified up to date in rodents, which is a big limitation for all the developmental studies. So we did the approach to try to overexpress the human protein, especially because we know that this protein interacts with FGF receptor 1, that is one of the main receptors for FGFs and FGF-2, and it is the one that is involved in most of the functions of FGF-2 related to oligodendrogliogenesis. So when we did it, first, we observed in the subventricular zone, as you can see in the left panels, a big disorganization, big masses of cells just under the subventricular zone, and the general disorganization of the primary cilia in the surface. But we didn't see something spectacular there. We saw some changes in the oligodendrogliogenesis in the different stages that we studied. And what was more important for us is that we saw a big amount of Ranvier nodes. That means that the internodal length of or segment in the corpus callosum should be smaller than in control conditions. And not only the number, but also the length, the size of the Ranvier node itself. And overall, measuring in vivo the conduction velocity between one side and another one hemisphere across the corpus callosum, we saw that the latency diminishes, so the transmission was larger because of all this accumulation of myelin. This was published some years ago in a couple of papers. And the next one, please. Just 2 or 3 years ago, the group of Fiona Deutsch identified a lot of quiescence neural stem cells in the subventricular zone that produces masses of oligodendrocyte precursor cells in the adult mouse brain. So this cell type has been named gorditas in Spanish. And the next one, please. We wonder if what we were looking at that time in this mouse were indeed these gorditas cells. We must come back to this in the future, especially since in the last times it has been reported that the oligodendrocytes that would be deriving from the neural stem cells in the subventricular zone and these gorditas, and the oligodendrocytes or precursor cells that are in the parenchyma are competing for the remyelination in some scenarios. The next one, please. So overall, it is very interesting what happens in the corpus callosum. That is what we published. when we overexpress anosmin-1, you see that we produce more OPCs. These OPCs didn't die and produce more mature oligodendrocytes, thicker myelin, and smaller Ranvier nodes, or larger Ranvier nodes, sorry, with a faster conduction velocity. But very recently, it was this paper was accepted last week. We've seen, among another different of studies, that it is not the case with the oligodendrocytes in the basal ganglia, and we see that in the striasomes, the overproduction of, or the overexpression of anosmin-1 does not reflect more cells specifically or any changes. And it's something that we are also studying in the cerebellum in collaboration with Annalisa Buffo in Torino. And again, we see some differences. What we saw at the very beginning that would be more myelin in the Purkinje cell axons at the early stages at P15, when we study that at P60, that is more or less the moment that myelination should be completed in normal conditions, there are no changes anymore. So it seems that all the myelin that we see in the cerebellum is not more myelin, but faster myelination in these conditions. What we are also completing the analysis now is that the overexpression of anosmin-1 also generates in the Purkinje cells dramatic changes in the Ranvier nodes, in the size of the Ranvier nodes that are larger and do not dynamically change during development. They remain in the presence of anosmin-1 very big, and also some changes in the initial segment. So the next one, please. With this, we have another demonstration that the activity of these molecules, or this kind of equilibria that we've described in FGF-2, FGF receptor 1, and anosmin-1, is relatively variable depending on many different circumstances. 10 years ago, we described in human tissue that in chronic active demyelinating lesions in multiple sclerosis in the brain cortex, the recruitment of OPCs, as you can see in the bottom part of the image, were done only by the attraction of FGF receptor 1 positive OPCs in the cortex. And the other OPCs that exist, FGF receptor 1 negative, they didn't respond, and they didn't try to remyelinate or to go towards the lesion, this type of lesions, to remyelinate. This was very intriguing for us. And the next one, please. Because it seems that for remyelination, there are some traffic lights, and this FGF-2 and anosmin-1 would be more or less defining when a scenario represents a possibility of remyelination, the active lesions, or the rim, or the perilesional rim in the chronic active lesions, but not the core of the lesion. In the chronic active lesions are, of course, the chronic inactive lesions. That is a scenario that is absolutely catastrophic to obtain recovery of the lost myelin. The next one, please. Of course, this is not the only actors, but it was very interesting for us to see how OPCs, all of them isolated from neocortex, behave in a very different way if we compare the response to FGF-2 and anosmin-1, as you see in the different ages of murine OPCs, A16, P0, P7, P15, and P60. there were some changes, and at the very beginning, what was more attractive, for example, that was anosmine, remains less attractive or even repellent instead of FGF-2 that remains more or less constantly as an attractive cue for this molecule. It's not only an attractive cue, but also FGF-2 increases the motility by itself of these OPCs. And what it was very interesting for us is that when we isolated OPCs from human biopsies, non-tumoral biopsies, we observed a kind of behavior in response to anosmine 1 and FGF-2 that was very similar to the one observed in more or less the mature mouse brains, that is FGF-2 attracting, anosmine 1 repelling. But if we studied OPCs isolated from tumoral samples, the behavior was absolutely the opposite. And both anosmine 1 and FGF-2 were attractants. And the behavior of this was exactly the same as the Hawk cells that are immortalized cells. So this is, in the next one, please. This is very interesting because we can, or we are aware that OPCs are much more heterogeneous than it was thought at the very beginning. And I think that our group has significantly contributed to the field. And this is something that now everyone more or less accepts in the field of remyelination. And this OPC functional heterogeneity relies on age, on the animal species, as we have also demonstrated, the embryonic origin of these OPCs during development, and also the presence of concomitant damage. That is something very interesting, especially for all these other omics and massive studies that should be taken into account. Slight changes in these conditions would reflect a very different result. Regarding sonic hedgehog, we did some experiments that I will tell you from the end to the very beginning. We isolated OPCs TD tomato positive from transgenic mice, and we injected that intravitreally. It has been done years ago, but the groups requested the use of a slight degree of inflammation for these OPCs to differentiate and form myelin within the retina. As you know, only the rabbits and very few other animals have oligodendrocytes within the retina, because when they colonize the optic nerve, they are stopped in the optic nerve papilla, and the OPCs do not enter the retina. So the intraretinal or intraocular segment of the optic nerve is myelin free, but is an axon that once out of the optic globe is myelinated, and is fully myelinated. So when we did this, we obtained some interesting myelinations, as you can see in the left panel on the left. But when we blocked sonic hedgehog with cyclopamine, sonic hedgehog activity, we get much more myelination. And when we agonized sonic hedgehog, we again came back to the more or less the control situation. And we replicated this using oligodendrocyte precursor cells isolated from a small flux mutant mouse or a small M2 mutant mice. The flux has a lower signaling of sonic, and a small M2 has an augmented signaling of sonic. And again, it seems that when we block sonic activity, there is more formation of myelin. When we have extra sonic activity, it is not affecting at all the spontaneous ability of the OPCs to form myelin. And what is important is that we've done it with zero inflammation extra. So we tried to have this model as a naive model. It's not exactly a control, because it is true that the intraocular segment of the optic nerve has not been, or it is normally not myelinated, because the myelinating cells are not there. But the rest of the axon, it is myelinated. So we consider that as a kind of naive condition. It's not a developmental condition because it's in the adult when we did it, but it's a naive condition. Again, the next one, please. On the contrary, if we go to development, and we see what happens with these two mutants with low sonic activity, the small flux, or high sonic activity, small M2, you see that there is some problem with the histograms on the upper part on the right, but when we have a high activity of sonic, we see a lot of OPCs, but lower number of major oligodendrocytes CC1 positive. That means that the highest signaling of sonic would arrest the OPCs in undifferentiated stages. And exactly the same happens in the bottom part, where you see that the corpus callosum, once examined, both from the expression of MVP and the presence of Ranvier nodes, the big change depends on the excess of sonic signaling and not on the lack of sonic signaling. This is normal development. The next one, please. What happens if we go to a demyelinating model like cuprison? We give cuprison for 3 or 5 weeks, and then we stopped, and we see the recovery in the second part of in the right panel. And in the left panel, we just identify the OPCs and then add EDU to see what happens with the proliferation. And in general, we see that, again, the overexpression of sonic signaling, small M2, resulted in a lot of OPCs with no affecting the MVP area at all in none of the conditions. But in the right part, as you see, if we induce the expression of T tomato during the acute demyelination, we see again that there are many more OPCs in the small M2, but also a normal number of myelinating oligodendrocytes with the presence of M2. And on the contrary, the flux resulted in lower numbers. There are no changes apparently in the thickness of the myelin that we obtain in the corpus callosum. The next one, please. In these conditions, and if we summarized, again, we find for this other old friend, sonic, that its activity depends on the different circumstances where it would be acting. Intravitreally, in the adult, that means in the naive conditions, the excess of sonic does not implicate anything, but the lower activity of sonic via flux, the small flux, resulted in more oligodendrocytes and myelin. This is very interesting. It's the same that we obtain also with cyclopamine. What happens during development in the young adult in a clearly developmental condition, if we eliminate sonic or we diminish the activity of the pathway, does not affect the development of myelin in these conditions. But if we increase the amount of sonic acting through smoothing, it results in a larger number of OPCs with a lower number of mature oligodendrocytes, as the OPCs would be kidnapped or arrested in undifferentiated conditions. And finally, in the demyelination phase of cuprison, it seems that the only effect that we have obtained clearly was thanks to the high signaling through sonic, and we obtain more OPCs, but no more myelin. And on the contrary, when we are acting on the active remyelination after the lesion, we obtain with low sonic pathway activity, low numbers of OPCs and low numbers of oligodendrocytes, mature oligodendrocytes and myelin, and the excess of sonic does not affect again the myelination process. So we should be very subtle when we analyze all these conditions. Why we say that? The next one, please, and I'm finishing. Well, this is just to tell you about another old actor that is osteopontin, that is very interesting for us in also as putative remyelinating actor, but I will not tell you this in detail. The next one, please. So of these cues that are interesting for us, at least FGF-2 and osmin, that is more or less a kind of system interacting with them, sonic, osteopontin, or with the small molecules or aptamers that we are using to remyelinate, is what we have interests. We see that human adult OPCs would be very important to be studied. The next one, please. First, because these are the cells that are present in the brains of patients, MS patients and other patients. And the second, because if you see the preclinical essays of the different clinical trials for remyelinating treatments, you see that some of them that have dramatically abandoned, what they have not checked properly is the preclinical studies. And this is very dramatic because they lost thousands of millions of euros or pounds or dollars, whatever, without doing proper preclinical studies, responding to these characteristics and heterogeneities that we see on the OPCs. And as a result, we have not a clear remyelinating agent available in clinics. The next one, please. And obviously, we have a limitation for the use of OPCs isolated from the human biopsies. First, because the size of the biopsies is smaller every day, fortunately for the patients. And the second, because the connection and logistics is not so easy. So now we are trying to develop directly reprogram or self-reprogramming human fibroblast into induced OPCs. And this is something that we reached at the end of last year, and now we are preparing the publication. And we have quite effectively obtained the protocol to generate this and try to use this as animal or in vitro models of disease, human in vitro models of disease. The next one, please. So, in summary, the formation of myelin is not so tricky. Cells do it in a massive way during development, but it is not so easy. And it's something that we try to replicate that in vitro, for example, or to replicate in a pathological scenario to treat patients is not so easy. And it's our dream to get this opportunity and to increase the arsenal to treat MS patients and other patients of primary demyelinating diseases and secondary demyelinating conditions with some effective remyelinating agents to combine with immune modulators, etc. The next one, please. And just to tell you who we are, the main actors of this work. The Sonic work was done by Sonia Noceira, that is now a postdoc in Jonah Chan's lab in University of California in San Francisco, and Miguel Marchena, that is a very serious postdoc in my lab since years ago, and some other people here that have collaborated in the FGF-2 and anosmin-1 staff. Some of them have already left the laboratory. So thank you very much, and ready for your questions.